In this video, I want to tie together a few different things to illustrate how the liver functions as a metabolic organ. And we're going to look at alcohol and how it's metabolized, and then we're going to compare it to sugars like fructose to show how they're similar and how they're different. So the first thing is when you drink alcohol or ethanol, it's all the same. Doesn't matter if it's wine or whiskey or beer or where the ethanol came from. It's the same molecule. And in your liver, you have two different enzymes. The first one, alcohol dehydrogenase, is taking that ethanol and converting it to an intermediate compound called acetaldehyde. That acetaldehyde then is converted by a second enzyme, acetaldehyde dehydrogenase, and converted into acetate. Now, the interesting thing is, if you have ever looked carefully at the alcohol content and calorie content of alcoholic drinks, you'll notice that alcohol has calories. That's because this acetate molecule, if you just add a CoA, cofactor A, it becomes acetyl-CoA, which, if you remember, is the first molecule that enters the Krebs cycle, and you can use it to make ATP. So ethanol contains energy, and what that means is, even though alcohol can be a poison, it does have nutritional content. You can get calories out of it. It's not the healthiest form of calories. But in order to make that happen, you have to have these two enzymes functioning. Now, what's interesting is the liver is where we see the most damage from over-consuming alcohol, but the liver is actually the organ that's getting rid of alcohol, right? And so if you look throughout the entire body of a person who was, a little drunk or intoxicated, you would actually see the liver is where the lowest ethanol content is. And the reason for that is that's where you find the enzyme. And since this enzyme is constantly converting ethanol into acetaldehyde, ethanol levels are a little bit lower in the liver than they are in the blood. Right? So how do you make sense of this? The liver has the lowest alcohol concentration of any organ, but it also experiences the most damage from alcohol. The reason for that is the acetaldehyde is actually the dangerous part. That aldehyde functional group is similar to formaldehyde. And so if you build up high levels of acetaldehyde, you're basically preserving the liver from the inside out. That's where the toxicity is coming from. Okay, so let's look at a couple examples. Imagine that a person had a minor defect in their alcohol dehydrogenase enzyme so that this step was slow. Still happens, but it's slow. If this person drinks alcohol, it's going to take a long time to convert the ethanol into acetaldehyde. The effect would be this person is going to be drunk for a really long time, and the blood alcohol will stay in their system for longer than it normally would because the liver can't remove it as quickly. On the flip side, if you imagine a mutation in acetaldehyde dehydrogenase, which is actually a very common mutation, really common in East Asian populations, and in this case, it's not converting ethanol to acetaldehyde that's the problem. It's converting acetaldehyde to acetate. And so in this case, acetaldehyde levels accumulate in the blood, and that doesn't make a person feel drunk. It makes them feel sick and nauseous, right? And so I'm using this to illustrate how the liver doesn't act like a filter. It's not filtering out alcohol. It's using enzymes to convert one molecule into another. And by the end of it, we get a molecule, acetate, that we can actually use. Now, for a lot of drugs, they don't have calories, and so for a typical drug, the end product would be something that gets filtered out by the kidney and leaves the body. Alcohol is kind of a unique scenario. All right. So, what impacts can this have? Well, interestingly, the data on this aren't entirely clear. What you end up seeing is if you measure how much alcohol people drink, like in drinks per day, and you measure their mortality rate, right, deaths per 100,000 people, what you'll see is an interesting thing, that the supposedly safest place to be is actually having about one drink a day is the lowest point on the mortality curve. And if you go up from there, mortality increases. But if you go down to zero, it also increases. And so it appears that a very small amount of alcohol every day is actually healthy or beneficial in some way. And the link appears to run through the cardiovascular system. So if you have one drink a day, your risk of dying from cardiovascular disease goes down a little bit. But if you start having more than 2 or 3 a day, your risk of this alcohol-augmented conditions really just means liver, cirrhosis, or cancer. And if you get over 6 a day, it starts to go up pretty dramatically. And so alcohol is a weird example of a molecule that seems to be mildly beneficial to one organ system, cardiovascular, but harmful to another organ system, liver. And it's possible to find this sweet spot right in the 1 to 2 drinks per day range where you seemingly can get the best of both worlds. All right, if you push too far, you'll start getting pathology. So alcoholic liver disease comes from a few different angles. In stage 1, fatty liver happens because the liver can only use so much ethanol at a time. And if the liver has enough calories and enough ATP, the alcohol has to just be converted to fat. And so all of these open white circles that you see in this tissue, those are deposits of fat or triglyceride that are filling up in the liver because it has so much ethanol to deal with, it can't all be used all at once. And this is how that ethanol gets stored. This is completely abnormal because evolutionarily, humans shouldn't have had access to enough alcohol to make this happen. Right? If fatty liver goes on for long enough, the tissue will get inflamed. And remember, the suffix itis just means inflammation, and hepatic means liver. And so hepatitis just means liver inflammation. This would be alcoholic hepatitis. Right? You can also get hepatitis from viral infections or bacterial infections or other toxins. And what you end up getting is a bunch of necrosis or cell death. And when those cells die, they can be replaced to a certain degree. But if that goes on for long enough and gets severe enough, instead of replacing the cells with new liver cells, hepatocytes, you replace it with scar tissue instead. And when scar tissue shows up, that's cirrhosis. Liver cells are gone at that point, and this is the stage where it becomes irreversible. So if someone stops drinking when they have fatty liver or even mild to moderate hepatitis, they can go back to having a somewhat healthy liver again. If you get to the cirrhosis stage, quitting drinking might make the progression slow down, but you can't replace that scar tissue with functional liver ever again. All right. Now, the interesting follow-up from this is, over the last decade or so, non-alcoholic fatty liver disease has actually become more common than the alcoholic version. And the strongest link to non-alcoholic fatty liver disease is a sugar called fructose in the diet. Fructose makes up half of table sugar. It's also enriched in high fructose corn syrup, which is what's used to sweeten a lot of sodas. And honestly, many, many processed foods now. Food manufacturers dump this stuff in everything. And to explain why, we're going to do a little bit of metabolism. All right, don't get lost in all the details here. The big picture is when you eat glucose, you have a few different options for how to use it. You can use the glucose to make ATP directly, right? Mitochondria, Krebs cycle. You can take extra glucose and store it as glycogen. That's fine. And if that's full, you can export it as fats. These VLDLs carry fats as triglycerides to store it in adipose tissue, right? So this would be adipose. Now, you know from the appetite regulation video, if you have too much adipose tissue, that should signal via leptin to your brain to decrease your appetite, and that should slow down consumption. And so eating glucose should self-regulate if everything's working properly, and the person will maintain a healthy weight, and their liver will be fine because the excess fat got sent to adipose tissue the way that it should, which we can regulate. All right. With ethanol, the problem is, one, you can use it to make ATP, but you cannot make glycogen. So there's no glycogen in the liver. And so as soon as you have more ethanol than you know what to do with, right, the next step is start converting it to lipids. Now, those lipids can still be excreted from the liver and be stored as adipose tissue, but since the cell is inflamed, a lot of them stay trapped in lipid droplets in the liver itself. Right? And for reasons that aren't completely understood, ethanol-derived fat does not stimulate the release of leptin, and so you don't get that inhibitory effect. In fact, what happens for some people is these double arrows are showing positive feedback, right, which is the substance of addiction. That's just saying the more you drink, the more you want to drink, and so the brain keeps motivating the person to get more and more. Right. In our last example, fructose sugar is a lot like ethanol. You can use it to make ATP. You cannot use it to make glycogen, but it still leads to inflammation. And when you have more fructose than your liver knows what to do with, because the liver is the only organ in the body that can actually metabolize fructose in significant amounts, just like alcohol, you get inflammation. That traps these lipid droplets in the liver. And so, same as alcohol, some is still exported to adipose tissue, but some stays trapped in the liver too. That's the fatty liver part. And also for reasons that aren't fully understood, fructose leads to leptin resistance, which means your adipose tissue still secretes leptin, but it decreases sensitivity in the brain so that the leptin receptors in your brain tissue start ignoring the leptin. You don't get that negative feedback effect, and the person eats more and more. This is why someone who has a very high sugar, high processed food diet is going to keep taking in lots and lots of calories and gaining weight, because it's not triggering the appetite control mechanisms that it's supposed to. Okay, so I hope you found this interesting. Fructose and ethanol are really very similar in a lot of ways. They can both cause liver dysfunction, and the only reason there's a major difference is obviously alcohol gives you the buzz effect, and fructose doesn't, and so it's harder for people to pay attention to how much fructose sugar they're consuming. All right, this was our last video for digestive, but follow along in the playlist, and we're going to move on to hearts and the cardiovascular system next.